Hello guys! So today in class we talked about the history of DNA, and tonight we're going to go into that a little bit more in depth. So first things first, imagine we're in the early 1800s. At this point, everyone's had microscopes for a while now, everyone's looked at the cells. The cool thing is to look through the microscope. So many people have seen the nucleus at this point, but the question was, what was inside that nucleus? So if you were one of these scientists, where would you start? First, before we get into this, tonight, as we're going through this, we're going to have these quick questions throughout the lecture. What I want you to do is write them down on a separate piece of paper. You can pause the video to write them down and then just hit play again when you're done. So question number 1 is, what type of cell has a nucleus? Number 2, what is a nucleoid region and in which type of cell can you find it? And number 3, which type or types of cells contain DNA? Prokaryotes, eukaryotes, both, or neither? So the first guy who said, I want to find out what's inside the nucleus, and he did it successfully, was Friedrich Miescher. What he was able to do was separate the nucleus from the cell and determine what chemicals made up the nucleus. And he did this through like a salt solution. What he found was there was a lot of nitrogen, phosphorus, and sulfur in the nucleus. He called it nuclein, but today we call them chromosomes. Basically what he discovered was the nucleus was partially DNA and partially protein. So that was a good first step, but what are chromosomes? Basically, it's how DNA is packaged inside the nucleus. Remember we talked about how there was 6 feet of DNA in every single cell in your body. Well, you know me, I'm pretty short. 6 feet is a lot taller than me, so how does that get into one of my cells? What you have here is the double helix, and that's the structure of DNA. And it starts to coil around these proteins, these circular little proteins called histones. And once they coil around the histones, those coil together, and then those coil together, until you have this X-looking structure. That is your chromosome. So basically, a chromosome is just DNA and the histones put together. That's how you can fit so much inside your cells. Well, at this point, we figured out what was in the nucleus. But the other big question was, what causes heredity? How do you pass your traits on to your kids? So, how do you have your dad's eyes or your mom's hair? This work was done by Walter Sutton. He was working with grasshopper chromosomes, and he found that all chromosomes come in pairs. You get one set from your mom and the other set from your dad. And you only pass one set of chromosomes to your offspring. Basically, he figured out that chromosomes are the basis for heredity. So that protein and DNA mix of the chromosome was getting passed on to your children. And something about it was passing on your traits. So, which 2 biomolecules are found inside the nucleus? What is a chromosome, and what is heredity? So that's great. We figured out what chromosomes are. They're DNA and they're protein. But which part gets passed on? Which part is the heritable material? Is it DNA or is it protein? That's what Frederick Griffith was trying to figure out. And he did this interesting experiment with bacteria and mice. There was 2 different types of a pneumonia bacteria. One that caused mice to get sick and die, and the other one that was harmless. The mice were fine. Well, he decided to take the disease-causing bacteria and kill it using heat. And when he gave that to the mice, they didn't die. It made sense. The bacteria was killed. But then he had the interesting idea of mixing the 2 together. The heat-killed one that causes the disease, and the harmless one. And when he did that, the mice got sick and died. Whoa! What he just figured out was that something inside the dead bacteria was transferring itself to the live bacteria. He called this transformation. But unfortunately, that still didn't answer the question, is it protein or DNA? So 20 years later, that's what Oswald Avery was trying to figure out. He purified Griffith's experiment, isolated each part, and found out that it was the DNA that was moving from one bacteria to the other. So DNA was the transforming material. But science likes to have everyone repeat each other's trials, repeat each other's experiments, because in this way you can really prove if something is true or not. Sometimes you can have fluke mistakes that give you the right answer once, but being able to do it many different ways is really going to make sure if you have the right answer or not. So Alfred Hershey and Martha Chase, they decided to test the same question, protein or DNA. They used a bacteriophage, and what that is a virus that infects bacteria. These are really great because they're only made up of two parts, DNA and protein, just like our chromosomes. So how did they figure out which part moved and which part stayed? Well, we had just entered the age of radiation, so they were able to use radiation to tag the elements in protein and DNA. Sulfur makes up a lot of protein, so they used radioactive sulfur to test for protein, and radioactive phosphorus to test for DNA because of the sugar phosphate backbone of DNA. What they found was that only the DNA transferred from the bacteriophage to the bacteria. Once again, DNA was the heritable material. If you look at this picture on the left, notice the glowing part is the protein shell. And when the bacteriophages try to inject their DNA into the bacteria, the DNA makes it inside, but the radioactive protein shell doesn't, because that's not what's getting passed on. When you have radioactive DNA and the bacteriophages infect a new bacteria, then they leave that radioactive DNA inside the bacteria. That's the heritable material. So why the controversy? Why was it so important that all these people prove over and over and over that it was DNA? If Griffith discovered transformation in 1928 and Avery identified it as DNA in 1944, why did they have to prove it was DNA again in 1952? Well, we did talk about how you need to repeat each other's experiments, but also scientists back then couldn't believe that DNA was the heritable material. It was so simple. It was just made up of 4 bases, whereas proteins came in many different shapes and structures and sizes. We're all complex organisms, so the thing that gets passed on had to be complex. It was hard for scientists to change their opinions back then because science is so social. You don't want to look silly in front of your friends, so a lot of times these scientists would publish their results saying, well, it looks like DNA, but I still think it's protein. So that caused a lot of confusion back then. Finally, once Avery, Hershey, and Chase had definitively proven it was DNA, the evidence was hard to ignore at that point. So now we know it's DNA. Well, let's start looking into DNA. What is it? What does it do? So Erwin Chargaff was the first that we're going to look at today. He took DNA samples from many different organisms and tested them chemically. He found that each organism had the same base ratio. What that means is, of the 4 nitrogen bases in DNA, adenine, thymine, cytosine, and guanine, the amount of A was equal to the amount of T, and the amount of C was equal to the amount of G. So that means if you had something that was 20% adenine, you would have 20% thymine. And put those together, well, that's 40%. You need to find out what the other 60% is, and that's 30% cytosine, 30% guanine. This led to Chargaff's rule, which means that A binds with T and C binds with G. So let's do this math really quick because you may need to do this on a test or some type of standardized test in the future. If you find out that your sample of DNA has 18% guanine, how much cytosine does it have? Well, Chargaff's rule says that C and G are the same and A and T are the same. So that means that our cytosine is 18% of our DNA sample. So how do we figure out the A and the T? That's a lot harder. We know that it's 18% G, 18% C, so we can add those two together. And you get 36. So 36% of your DNA is C and G. If we subtract 36 from 100, because nothing can go above 100%, you're left with 64. So 64% of what's left over is going to be adenine and thymine, but those have to be equal. So that leaves us with 32% being adenine and 32% being thymine. You just divide this by 2. Now, it's obeying Chargaff's rule, and you're getting 100%. So, kind of trick question down here at the bottom. What percentage of uracil does this particular organism have? Well, in this case, for DNA, it's going to have 0. Uracil is part of RNA, not DNA, so you'll never see it in your Chargaff's rule for DNA percentages. So let's move on to what DNA actually looks like. In 1952, Linus Pauling was also hot on the trail for the structure and chemical basis of DNA. But he thought that it was proteins. He thought proteins were the heritable material. He discovered that proteins had something called a helical shape. But once he figured out that DNA was the heritable material and not proteins, he decided to apply this idea to DNA instead. And he created a model of DNA that was a triple helix. Close, but not quite. His discoveries were going to help the next scientists make even bigger breakthroughs. Rosalind Franklin and Maurice Wilkins at the same time were using X-ray diffraction to take pictures of the structure of DNA. And their pictures pretty much said that DNA was a helix, but it was a double helix. Right down the hall from them, at a Cambridge lab, was James Watson and Francis Crick, and they were the guys who eventually solved the puzzle. They took the information from Linus Pauling, from Chargaff, and from Franklin and Wilkins, and they put it all together to create the structure of DNA that we now know. The double helix with the bases that bond to each other. Here it is now. You have the backbone that's shaped like a helix. There's 1, and there's 2. And then the bases are bonding with each other in the middle. A bonds with T. Oh, sorry about that. A bonds with T, and C bonds with G. So that obeys Chargaff's rule. We have our helical shape, and the bases are bonding to each other, so it's a double helix. Pretty cool. Quick questions. What do you think were the 3 most important discoveries that helped Watson and Crick discover the structure of DNA? And 2, how is the discovery of DNA a social activity? You can pause and answer those questions and join us on the next slide. Now that we've known the structure of DNA, genetics has just exploded as a new scientific frontier. And in 2003, the world succeeded in mapping the entire human genome. What that means is we were able to figure out all of the little letters that make up our DNA, A's, T's, C's, and G's, and what order they were in. You think, why did we have to do that? Well, it's helped us do things like fight cancer and diseases and genetic disorders, and it's really opened up a whole new world into how we see ourselves and medical breakthroughs from this point on. So that's the history of DNA so far. I hope you guys enjoyed it, and we will talk about the structure of DNA tomorrow in class.